thanks everyone for attending the Demand seminar. It's going to be a very short introduction to Michael Firbank, because obviously Michael's known by quite a lot of you. He's one of our senior imaging analysts, and has been involved in a lot of projects over the years, particularly on the imaging MR side. And today he's going to be speaking on some of the results of the CAPfield study, taking a look at the functional MR in patients with DOB and also Parkinson's disease with dementia, and telling us a little bit about some findings as part of the CAPfield, particularly in regard to attention. Okay, Michael, over to you.
Thank you, John. So as just introducing, we'll be talking about the fMRI results from attentional. I put this slide in just on the off chance that anyone doesn't know about Lewy body dementia, but as probably you all know, Lewy body dementia is responsible for about 10 to 15% of late life dementia, and symptoms of fluctuations in cognition, Parkinsonian motor features and visual hallucinations, which is in distinction with Alzheimer's disease, which is the most common form of dementia, which principally has memory loss as a clinical feature. Attentional deficits are particularly common in Lewy body dementia, though they are also present in Alzheimer's disease. This is a slide from a study of over 10 years ago by Clive Ballard when he was here, looking at a digit vigilance task where people are shown a stream of numbers, and you have to press a button every time a particular number appears. And the control patients are in white, Alzheimer's in black, and dementia with Lewy bodies in grey. You can see that the dementia with Lewy bodies miss a lot of the target due to attentional lapses. And so the Alzheimer's don't do too badly, but particularly for more advanced Alzheimer's, they also suffer from attentional deficits. And it's thought that these lapses in attention may have a role to play in visual hallucinations in dementia with Lewy bodies. So as well as being interesting to understand their own right, they may shed some light on that aspect. So as mentioned in the introduction, this is part of a study we did over a few years called CAPfield, Cognitive and Attentional Function in Lewy Body Disease. CAPfield also happens to be the name of a small village in Norfolk, which looks very nice. It's a village sign, according to Wikipedia at least. So the aims of the study was basically to do an attentional task and look at performance on it in terms of reaction times, etc., but also to do this during functional MRI, so we could get some sort of idea of what brain activity was going on, to see if we could pick up any differences in brain activity between the different dementias. There was also some EEG done, but I wasn't involved in that, so I'm not presenting any results of it. And this was in a group of Alzheimer's disease, healthy controls, and Lewy body dementia, which was combined dementia with Lewy bodies and Parkinson's disease dementia. So there are a number of different ways of looking at attention. We used a well-established model developed by Posner and colleagues, which basically divides attention into three components: that of alerting, which is basically being aware of any changes in the environment; orienting, which is sort of selecting relevant features out of the environment, such as something happening over there; and executive control, which is basically conflict resolution and decision making, so choosing a particular stimulus that you want. And Posner developed a task in order to probe these three components of attention, called the attention network task, which we slightly adopted and used in this study. So during the task, people were basically looking at a computer monitor, which is a black screen. In the center of the black screen, there would be three small boxes and a cross in the middle of the central box. And after a variable period, sometimes a cue would appear. So sometimes there was no cue, and no change. Sometimes there was a neutral cue, so the central box lit up to indicate that something was about to happen. And sometimes there was a directional cue which told people not only that something was about to happen, but where it was about to happen. So that lasted for 200 milliseconds. It went back to the black screen with the grey boxes. And then a target appeared, which was 4 arrowheads. And the task people had to do was to decide the direction that the majority of arrows were pointing in. So these arrows were either congruent, so the arrows always appear in the box which was indicated by the directional cues. The directional cue was always valid. So the arrowheads were either all pointing the same direction, which was the congruent condition, which was fairly straightforward. You look at it and it's immediately obvious which direction. Or incongruent, in which case one of them was pointing in the opposite direction to the other 3. And this incongruent arrow was either the end arrow, which meant that you've got 3 arrowheads pointing in the same direction and one pointing in the opposite direction, which was an easy incongruent situation. Or, in order to introduce a bit more conflict in the task, we had a slightly harder situation where one of the middle 2 arrows was pointing the opposite direction. So you've got one arrowhead pointing left, 1 right, and then 2 left. So, and this was repeated several times with counterbalanced combinations of cue and target. So Posner basically used this to estimate, get a measure of his 3 components of attention. So the alerting effect was got by comparing the no cue with the neutral cue situation, just to see how much quicker you are when you know that something's about to happen. The orienting effect by comparing the directional cue with the neutral cue, so what's the added benefit of knowing in advance where something's going to happen. And his executive control by comparing the incongruent with the congruent target to see what reaction times in those 2 situations are. So people were doing that. We trained everyone to do this outside the scanner, and then they repeated it whilst the fMRI scanner was running by viewing it, so they're sort of lying in the scanner with a mirror above their head so they can see it on the screen. So I'm just going to give a very brief overview of how fMRI works, just for people who are less familiar with it. So what we're measuring is a thing called BOLD, which is blood oxygen level dependent signal. And this relies on the fact that when a particular area of the brain is active, it increases its oxygen consumption. So the oxygen is provided by hemoglobin in blood. So the brain increases the blood flow to that region in order to supply more oxygen. However, it overcompensates, so it actually provides more oxygenated hemoglobin than is required, which means that the net result is both an increase in blood flow and an overall increase in the amount of oxygenated hemoglobin. Now this is important, this contributes to the MRI signal because deoxygenated hemoglobin and oxygenated hemoglobin have different effects on the magnetic field. And the net result of this basically is that the decreased deoxygenated hemoglobin leads to a rise in the MR signal. It's quite a small rise of about 1% on quite a noisy signal. This is it in sort of schematic form, so you've got a rise in neural activity, which is followed by a rather delayed and more broad increase in the blood flow. This is, however, broadly proportional to the magnitude and duration of the stimulus, so 3 curves here from increasing amounts of stimulation And basically the analysis is performed by looking at the signal at each point in the image, looking at the time course of the signal, and finding pixels where the change in signal correlates with the timings of the task. So if the signal increases every time a target increases, you know that signal is active in the task. So fMRI has been used to investigate a whole range of cognitive functions, and has shown different cortical regions involved in various aspects of brain function. Two simple examples here is if you're looking at a visual stimulus, but this is looking at a blank screen, you find activation in the occipital lobe in the back of your brain. If you're doing a motor task, so moving your hand versus being at rest, you get activation in the motor cortex up at the top of your brain. And different tasks will engage different cortical regions. But it's also been observed for some time now that there is a pattern of cortical regions which show a deactivation during a range of cognitive tasks. So visual search tasks, or numerical calculation tasks, or attention tasks. This network of midline parietal, midline frontal, and lateral parietal typically shows a deactivation during the task and then an increase at rest. This was first described and sort of named by Reichel in PNAS publication of 15 years ago. And he called it the default mode network because this network of cortical regions is more active at rest. So he thought, well, that's the brain's default state. And it's associated with the activity in this region, or at least the function of this region seems to be involved in self-referential thinking, memory, thinking about the past, planning the future, and possibly daydreaming. But it has also been observed that it shows that this network of regions shows similarities with some of the regions which are affected in Alzheimer's disease. So there's a publication by Buckner 10 years ago in Neurobiology of Diseases, where he pointed out that if you look at glucose consumption, metabolic rate of glucose consumption in Alzheimer's patients at rest, they typically show reductions in midline parietal, lateral parietal. And if you look at the distribution of amyloid, the neuropathological marker for Alzheimer's disease, this also shows this midline parietal, lateral parietal, and also slightly more frontal changes. But he speculated that there may be some link in either changes in this default mode network cause the amyloid increase, or vice versa. It's also been noted that when Alzheimer's patients are doing a fMRI task, they show fewer reductions in fewer deactivations in this network compared with controls, which may be because their blood perfusion is quite low in those regions in the first place, so it can't deactivate because there's not much going on in the first place. However, we were interested in the Lewy body dementia, and Lewy body dementia and Alzheimer's disease show relatively similar patterns of glucose uptake. So this is from a study we did a couple of years ago. This is the Alzheimer's and the dementia with Lewy bodies below. So the dementia with Lewy bodies show more decreases in the occipital cortex, but in terms of the parietal lobe, they're relatively similar, except you can except that. So this is now a sagittal view. So this is the sort of bright midline parietal that you see in controls, showing there's a high glucose consumption in the controls in this region. The Alzheimer's show basically very reduced glucose metabolism in that region. The DLB showed preserved part of it, which Lim and colleagues described as the cingulate island, this region being the posterior cingulate cortex. And this is an important node of the default mode network. So one question we wanted to know was, given that this small part of the default mode network is preserved in DLB, how is that going to affect the fMRI results? So the questions we basically wanted to get out of the study were, first of all, how the reaction time and behavioural responses are affected by the different cue and target conditions of the attentional task. From an fMRI point of view, whether any regions of increased or decreased activity which might relate to those behavioural changes. Given the previous research, we expected the default mode network to be abnormal in Alzheimer's, but weren't sure what to expect in Lewy body dementia. So we had recruited 23, well, I didn't, but Alison and Jean-Paul and other people, recruited 23 control subjects, 23 Alzheimer's, 19 dementia with Lewy bodies, and 13 Parkinson's disease with dementia. Now, Parkinson's disease and dementia with Lewy bodies show very similar cognitive symptoms. The diagnostic classification is basically if you've had Parkinsonian motor problems for more than a year, you are classified as having Parkinson's disease with dementia, whereas if your dementia is more recent than your Parkinson's problem, your dementia with Lewy bodies, there's been much discussion about whether they are one disease or one spectrum of disease or two separate disorders. We grouped them together as one disease. We did also look to see if there were any differences between them in terms of the results, and there basically weren't, so felt justified in grouping them together. So just a very brief summary of the actual task. People did 46 runs of the attentional network task in the scanner with 36 trials on each run. We excluded any run with more than with fewer than 2/3 correct responses, because people may have been guessing on those, and just the scanner details, etc. So, some results. So the groups were pretty well matched for age and sex and dementia, disease of duration of cognitive problems. As expected, the Lewy body dementia had more motor problems, but the dementia groups were well balanced on their MMSE and CAM-COG, so they had similar cognitive function. So these results were all from our recently published human brain mapping article. So in terms of their response on the task, I think we excluded five Lewy body people because they didn't manage the task sufficiently well, but 32 people from the Lewy body dementia group did manage it, and 23 Alzheimer's managed it. So the task was pretty well managed by people with dementia, with people responding to virtually all trials for the controls. Two dementia groups responded to about 96%, so they did have slightly more missed trials. In terms of correct responses, again, the controls got 99% right, Alzheimer's got 95%, and Lewy body dementia 91%. So the Lewy body dementia people did do slightly worse in terms of correct responses. The fMRI only analysed, or at least we only looked at the correct responses. So in terms of comparing the different components of the attention task, shows the reaction time for the no cue on the left, neutral cue and directional cue for controls AD and DLB. And the most obvious thing from this graph is that the Lewy body dementia patients were very slow, and the Alzheimer's were slow, but not as much so. So But in terms of difference between the actual cues, there was not that much. So this over here is showing the difference between the no cue and the neutral cue. So the control group did have faster reaction times by about 30 milliseconds for neutral cue versus no cue, which was significant. The two dementia groups didn't show any benefit of cue, but there weren't actually any group differences, so there weren't any differences between Alzheimer's and controls. The directional cue was a benefit to all people, so everyone was faster on the directional cue than the neutral cue by about 75 or so milliseconds. But again, no differences between the groups. In terms of reaction times to the different targets, so again, in keeping with the previous ones, the Lewy body dementia are the slowest group, followed by the Alzheimer's and the controls. Everyone was faster to the congruent, slower to the easy incongruent. The interesting thing about this, so this is a graph comparing the congruent versus the easy incongruent. So that's easy incongruent is with the end arrow pointing in the opposite direction. So the Lewy body disease group were slightly slower than significantly so at least, than the control group. The main difference was comparing the hard versus easy incongruent. So the controls didn't find the hard task that much harder than the easy task, whereas both of the dementia groups did. So they had a much more increased reaction time for the hard task versus the easy incongruent. But there weren't actually any differences between the two dementia groups on that. So to summarise that, the Lewy body disease group were slower than the Alzheimer's, who in turn were slower than the controls. The alerting effect was only significant in the controls, but there weren't any group differences for any of the cue things as such. The oranging was similar. The difference between the congruent and incongruent was similar, but as the tasks got slightly harder, that was felt to be more noticeable in the dementia groups than the controls. So the harder task was harder for the dementias than the control group. So the MRI findings, the scanner downstairs with two of the radiographers showing how it works. So this is showing the activation to the cue, the neutral view versus no cue, and basically all groups showed this increase in sort of occipital areas and parietal areas to the cue versus the no cue, which makes sense because those areas involved in visual processing. But basically there weren't any differences between the groups. All three groups responded similarly to the cue. The directional cue was also similar, they all responded fairly similar, which is in keeping with the fact that we didn't see any behavioural differences on the reaction time for the cues. Comparing the response to the target, however, was slightly more interesting. So for the congruent versus all of the incongruent trials, the controls show this activation in the frontal and parietal areas, and show in blue this deactivation of the default mode network. The Alzheimer's group show a similar pattern of activation, but no deactivation at all. And the Lewy body dementia basically looked very similar to the control group. We didn't see any regions of increased activity anywhere, and showing that the cortical regions involved were all pretty similar between groups. Comparing the hard versus the easy incongruent task, where we saw those reaction time differences, basically the controls don't show any more activation for the hard versus the easy, which is in keeping with their reaction times not increasing by much. The Alzheimer's group, however, do show activation of a number of regions involved in the task. So when the task gets harder, they seem to increase their activation in these cortical regions, which the Lewy body dementia group did also, but not to the same extent as the Alzheimer's group. So basically, we didn't find any abnormal increases in activation, so there was no extra cortical regions being sort of used abnormally. The Alzheimer's group showed no deactivation and increased activation of the parietal and frontal regions, which might be compensatory, or to try and compensate for their sort of loss of brain tissue. The DLB group showed fairly normal-looking default mode network deactivations and slightly increased activity, but not as much as Alzheimer's. So we just wanted to have a look at this deactivation in a bit more detail. So drawing a region of interest to extract the mean signal from this parietal midline region. This is showing the activation in the different target conditions versus rest, as opposed to differences between the target conditions. So the Alzheimer's, as I've said, showed no deactivation at all for any condition. The controls don't really show any deactivation for the congruent, which but increasing amounts of deactivation as the task gets harder. Whereas the Lewy body dementia group show this deactivation even for the congruent situation, which increases as the task gets harder. So one thing I was pondering about on this was whether this was due to them spending more time, as it were, so the activation is greater because the total reaction time is longer. So this graph is going to show the reaction time for each individual versus the fMRI signal. So these are the control responses to the congruent target, showing overall no no deactivation, but possibly a trend for longer reaction times to be associated with greater deactivation. If we add on the incongruent trials, there's a definite trend with the longer reaction times having more deactivation. And if we look at where the Lewy body group are on the same graph, we can see that basically the Lewy body congruent responses are about the same time as the controlled incongruent ones and about the same magnitude of deactivation. So perhaps it could be that during the interval between the target appearing and the response, the dementia Lewy body group deactivate their default mode network whilst they're thinking about it, and the total magnitude of this deactivation is determined chiefly by the time they're spent thinking on it. So that was the deactivation from this midline region. What does it look like if you look at the activation in one of the parietal regions, which is activated during the task? And basically, the activations for the dementia Lewy bodies group look about the same magnitude as the controls. It's just that they happen later. So the parietal activations are not increased purely because they're spending more time on the task, but they are increased when the task gets a bit harder. So this is a bit of a difference between these two. So the reaction time is increased, and the default mode network deactivations are also increased, seemingly in proportion, whereas the cortical activations involved in doing the task are not really increased compared to controls. So there are three possibilities I was going to suggest as causing this. So this is the schematic of the normal situation where you've got the bold brain activation on the y-axis and reaction time on the x-axis. So the parietal region is activated during task and then goes back to normal, and the default mode network deactivation occurs during the same period. So one possibility was that the Lewy body group just take longer to respond because they've got motor problems. So the parietal region, which is involved in coming to a decision, manages to do this as quickly as normal, and then there's just a delay whilst this is translated into a motor response, and during the entirety of that the default mode network is deactive, which, let's say, fits with the known motor features. On the other hand, if that was the case, we might have expected to see some increase in motor activity in the motor cortex or the basal ganglia, which we didn't really see. Another possibility is that the deactivation occurs pretty much as soon as the target appears, but it takes a bit of time before the parietal networks are engaged. But when they are engaged, they again act normally. And again, that there is some prior knowledge that Lewy body patients can be difficult to disengage from one thing to do to another. On the other hand, we didn't see any increase in the occipital activation, and you would have expected that the visual cortex would have to be active in order for people to notice that the thing was there. But again, it's a possibility. Or the third possibility is just that they're basically slow. The deactivation in the default mode network is the same magnitude as the controls and longer, whereas the parietal network is inefficient, so it doesn't activate as much, but it does so for a longer period. So the overall height of this curve is not as great as the height of the normal curve. So, just to conclude from all that, we found that the reaction time was slowest in Lewy body dementia, with Alzheimer's being intermediate between controls and brain activation was highest in the Alzheimer's group, and the default mode deactivations were increased in Lewy body dementia but absent in Alzheimer's disease. So we definitely saw differences between these two dementia groups. And possibly I'd suggest that in Lewy body dementia, the cortical regions, perhaps due to poor connectivity between them, are less efficient, which leads to slower cognitive processing. And then the Alzheimer's group, possibly due to amyloid pathology, there's, the default mode network is basically not working properly, and there is some increase in cortical activity which might be compensatory given that their reaction times were not as slow as the dementia with Lewy bodies. So that's it really, so thank you for your attention. I'd just like to thank John Paul, who was the PI in the study, Alison, who interviewed an awful lot of people and performed the attention network task and was probably Paul sick of it. The ML radiographers who always do a marvellous job in getting people to lie still for the scanning, and Zina came over from Germany in order to work on the data. And, thank you.
Thank you, Michael, for that interesting and very clear talk. Does anybody have any questions? Raj.
Thank you, Michael. Going back many years, again, time when Roseanne Kenny and Clive Ballard were around, we looked at reaction times in the post-stroke survivors. So I wonder about specificity. Reaction times were longer. It did separate the two groups, the declined and the stable group. And so I wonder about specificity in terms of how would you place stroke group in,
Um,
in your scenario with DLB, AD, and the controls?
Yeah, I mean, I don't think reaction time is going to be a sort of diagnostic test as well. I mean, not going to be supportive, but I guess in the post-stroke, you definitely know that you've got disrupted connections between brain regions, due to the infarct. So I would perhaps they would be slightly more DLB-like in their overall reaction time. I don't know. We have also been looking at in the group other aspects rather than just looking at total reaction time, looking at variation of reaction time and how it differs from trial to trial, which might be more specific to dementia with Lewy bodies in terms of they might have more variability.
I think we did try to separate those individuals who didn't have obvious cortical infarcts, but had mostly sort of subcortical small vessel disease type of lesions, and they showed. So I mean, yeah, probably disruption of the subcortical circuits, maybe frontal subcortical pathways. But I always wonder about what particular pathways, if we can define a little bit better the bundles or the cortical connectivity. I think that would be, a way forward, at least a way forward, or at least an advancement to where we are in terms of.
I mean, I'll just say that. I mean, certainly the original idea of the Posner model is that the alerting is perhaps subcortical, the orienting is perhaps a parietal-based activity, whereas the executive conflict one is more frontal. So there might be ways of teasing those different elements out in terms of attention. But I think it is slowing of information processing, disconnections, which are probably maybe driving the vascular. But it would be interesting to see. I mean, I don't think anybody's looked at this particular task in vascular disease at all. I haven't seen anything.
I haven't seen anything.
Strokes, but I mean, it's an interesting question to look at.
What did you think of the ANT task? I'm curious. Compared to
I'll let you go first.
Compared to other models of attention that you could have used to probe the system.
Well, I don't know about as compared to other models.
Or to tests that, let's say, that would load slightly differently on hypothesized.
I mean, it was slightly surprising that we didn't see more influence of the cue. We did ponder after the event that so some studies which have used it to have had invalid directional cues. So you get a faster response when it's when it's correctly cued, but on about 10% of trials the directional cue is in the wrong place, and that much increases the reaction time, and that might have been an interesting thing to do. On the other hand, it might have confused people.
I mean, I think the ANT has certainly had good face validity, and also I suppose one of the attractive things was that it could be done by people who are relatively impaired, because it is actually a very simple, independent of memory. It's quite a working memory, executive-based task. So that made it quite appealing from this population, and we got really good response rates compared to a lot of other tasks which people try and do. And the more complex you make these attention tasks, you get a sort of complete failure, and then you have a floor effect and then an unusual group who participate. But I think, we were surprised by the lack of cueing effect, particularly orienting, because we hypothesized that that's meant to have a strong cholinergic parietal basis, that yes, we would actually differentially see an effect. And some people have seen some things in other studies using ANT, and a lot of it may also come down to the mechanics of how long the cue target interval is. And we've slightly modified our task as well to make the executive a little bit harder, because when we initially did some pilot work with the scanning, we weren't seeing huge amounts of activation. And I have to say, honestly, some of the original studies with the imaging were maybe a little bit overhyped. and it's only when we got into that we sort of discovered these things. So, it has some benefits, but I think there is some limitations on this task.
I think the other thing was partly because we were doing an fMRI study. It's a visual cueing, which is potentially a problem because the interference between the visual problems in the Lewy body dementia group and any attentional effects.
We know that well. I guess from my own work, we know there's such a strong response to visual cues and almost an inability to inhibit that response. So it's
response inhibition or switching attention is probably, those sorts of paradigms might potentially unearth a little bit more than this one did. So I think, for example, the orienting one, as you've described, or an invalid cue, might be quite a good one to tease apart the groups better.
Can I ask one more question? So when you're reporting your behavioural data and you have your imaging data, how do you factor in the error rate on the behavioural data? So we've got the times, the response times, but they'll get it right or wrong. So how's that accounted for in the analysis?
Well, we basically accounted for it by just looking at the correct responses. And the correct response rate was quite good. But yes, we didn't do a sort of trade-off between accuracy and speed.
Thanks.
We did model the reaction time in a variety of different models of looking at the bulk response to see if that had an impact, and it was all very complicated, but overall actually the very more simple modelling actually came out better.
I mean, in terms of, I don't think there were any, the error rate data looked quite like the response time data, so that the dementia, Alzheimer's disease got slightly more wrong than controls, and the Lewy body dementia got slightly more wrong than them. So it followed broadly the same pattern. So I don't know if there would be that much more to.
Although perhaps more stronger activations or deactivations in the case of the DLB from the default mode network, the incorrect responses.
Possibly. There weren't that many incorrect responses.
Chris?
For your LBD cases, you've got a mixture of DLB and PDD. Is there any relationship between PDDs having more severe changes compared to the DLB? Just in terms of this being an attentional test, and if you think that the cholinergic system is more likely to be depleted in the PDD cases, or worse depleted than, say, Alzheimer's disease.
Yeah. We did look at the behavioural and neuroimaging responses to the between the DLB and the PDD, and didn't see any differences basically. They were all scanned taking whatever medication they were taking, and I think there might have been some good differences in cholinesterase.
Well, no, not so much cholinesterase inhibitors, but certain dopaminergic.
Certainly dopaminergic was good.
Or in the PDD group, of course, but that didn't have an influence on the reaction time. Particularly, did it?
I don't think so.
Yeah, there was a major influence. Lewis, I don't know if you want to comment. You've looked at the same cohort from a resting state MR point of view, comparing PDD versus DLB. I don't know if you wanted to say anything on that.
Well, in the resting state, we didn't see any differences. Very mild, not so easy.
Yeah, so there was much more commonalities than there was differences between the two. I think the PDDs had slightly more fluctuations, if I remember rightly, than the DLB group on the clinical scales.
I think they were a bit more depressed as well.
A bit more depressed.
Is the PD in that group relatively long-standing PD?
Yeah, I can't remember offhand the duration of the Parkinson's disease, but yeah, they were chosen. We tried to avoid people who were sort of in that slightly grey area, but actually with established PD developed dementia subsequently. So. other questions? I've just got one question, Michael. How are we going to improve those models? Maybe that's a question to take away from this.
Yeah, well, if the EEG showed anything, that'd be nice.
Okay, well, we'll maybe get Ruth to talk about the EEG and bring that to a meeting. Any final questions? Otherwise, I think we'll wrap up. Oh, sorry. Liz.
Sorry, no, is it possible to use DCM, dynamic castle modeling, to test the slight delay in hypothesis?
I guess it might be.
DCM, dynamic causal modeling, that's one of an imaging technique to take a look at causal relationships between activation in one area and another.
Moderation, so.
Yeah. So if you see activation in one area, then does that drive at a subsequent time point activation in another area? So maybe you might be able to see, for example, the default mode network deactivating first, and then you see the parietal cortex, shall we say, as part of the task-based network coming alive, or vice versa. Okay, thank you very much.